Morning.
Morning.
Good morning. How are you guys?
Hi, how are you?
Excuse me.
I'm doing okay.
All right, there's a Notion page. Thanks once again, Jesse, for bringing that up. Morning, Morgan. Any updates you want to start with?
I guess I can start with a few things. I mean, my general update is it's finals time. I think I have one homework assignment left, and then it's a final project and a final exam. That's what's up. There's some hurdles I had to jump through for some other things, which I mentioned elsewhere, but things are going well. I'm really shifting into what is ahead zone. Um, it's been a kind of like it's been a little bit of a disappointing last few months, but also I'm trying to let myself adjust to grad school and adjust to other things that were going on. But that's okay. I'm putting some things together for what's ahead, and I'm looking forward to that. In a little bit more concrete terms, yeah. We had Cognition Futures meeting, which was quite nice. We basically have worked our way through Is the Coding a Relevant Metaphor for the Brain paper by Brett. And we're kind of looking at potentially some of the critiques next time. And it's a really great topic and a great question because his arguments basically the coding metaphor works in one particular way, but there's like three different aspects of it, and saying I don't think causally the structure matches. And the conclusion of the paper, if I'm remembering things this early in the morning, which I think I am, was sort of like, he's kind of making these light the paper is basically, critiquing that idea and then loosely gesturing towards some other work that like, what's the alternative? And that's kind of where we were and trying to figure out what his alternates are, looking at some of the references. Since I have it up here, I'll show this quickly. Here we go. So at the end of it, we were talking about these other references, like what could cognition be if not computation, or connectionism, or dynamic systems, which I have not dived into that paper, but like, I think that's sort of, the conclusion of all of that is going to be this. And I'm curious to go through the responses to see what else is there. I mean, he talked about it incorporating sensory motor lobe. He talked about this other, causal circuit explanations and these other big citations. I think this was the last citation in the paper was the Gomez-Marin one. And they'll mention process philosophy and these other things here. So I mean, it's been nice. That's been very constructive, and I think in my perfect mind, like having an year-end summary of like where a Cognition Future is, that'll be good. I know in the past we've done like open house things or like made material for updates, and I don't know if we want to. I haven't thought about that. It almost makes me uncomfortable to say because like, ooh, I don't like what would that entail? But I think Cognition Futures is in a nice spot, and I'm very appreciative for everybody's efforts into that. And I think we are kind of at a place where yes, we can maybe put together a position paper about what we're looking at. Where we can push some things in a little more concretized way now that we're here, and we've had some experience at these different events, and to be talked about later is sort of the upcoming. I don't want to. I want later on today. I do want to get to like that the deadlines board. I don't know timeline because there's a few things I want to add to that. So that's sort of that realm. There are things with Joe Pro that I've been working on, but obviously everything's a little bit. It's just not going to be a time period where I get to push a lot of stuff forward. So hopefully after the next in about, I think my window is like 6 or 5 days from now. I will be in a brief winter break. I'm getting used to how California does their trimesters and stuff because it's different. I actually really like the shorter 10-week periods than like 14, 15, 16 weeks semesters. I think it's much better to have that shorter time period. At least from a student standpoint, I wonder if, as an academic person trying to go in depth on a topic, I would disagree. But I, in terms of the content and the pacing, I'm finding the 10 weeks really appealing, even though it's radically different for me. But anyway, outside of that, I'm kind of juggling between student hat and Cognition Futures hat and then lab admin hat. But basically, a lot of year-end stuff is here. There's a few events I'm interested in looking at it. We'll talk about later when we talk about future events, things to submit to or things to think about. And like I've been saying recently, building out another sort of project to I guess umbrella project to incorporate some things that's in the works too. Let me look at one thing before I step away from the mic, because I just wrote a little summary here. Oh yeah, Bradley. Someone reached out to me with an email like stating interest in the lab, and I think I forwarded that to you. Did you get that?
I did not. I didn't see it
Okay.
in my inbox. So
I will send that to you. It was someone they just like it was kind of a cold almost application with an email and it's kind of a cover letter.
Right.
I'll forward that to you again. It was kind of a random thing.
Okay.
So we have a few people interested in the lab as usual, but I want to kind of make ourselves a little presentable for the next batch of people coming through next year. So that's on the docket, and I think we can talk about that today, I suppose. But at some point, I would like to have a specifically administrative focused meeting on preparing for next year. That's it from me, I suppose.
Yeah. Well, thanks for the update. Yeah, I think we should get started for 2024. We'll talk about that a little bit today, but yeah, and please send me that inquiry. But yeah, I think that's good. We had kind of dropped, or like last year, we did a bunch of videos at the end of the year. It was the Open House 2022, and so we have some good videos in that playlist. But we now, I'm wondering if we should update it for 2023. And I have one up there already. It's the devorm update for 2023, so that's up there now. But we can still make other ones if we want. I don't want to spend too much time on it, but I probably will do an end of the year recap. So anything else you want to do, if it's a video that we don't have, like some new project you want to start, or if it's something that maybe we'll do a Cognition Futures recap too. That would be good.
I mean, if I could just I was gonna say if I could just wave a magic wand, but then I was like, if I could just easily engineer a chat GTP4 prompt that would create, chat GTP, make a 5 to 8 minute set of videos for all the different projects in the lab, reviewing briefly things that happened this year and an outlook for next year, and press the button. And I would love to do that. I think the more realistic version of that right now, no commentary on whether or not AGI is here or not, is kind of just like doing like just recording me, whoever, like for Cognition Futures or whatever, saying here's what it is: the Cognition Futures, the cybernetic stuff, And I don't even know, like some of the inhibiting questions for that are like, do you want it, like, is the project going to be called, like, reimagining cybernetics, or we have the cybernetics reading group now that's developed from the other reading group? Like, I don't know how to break that down, but like just like basically flash talks. I think like flash talks for all the projects would be, even if it's sort of informal, not perfectly structured, a couple slides. Like, I'm not keen to make a lot of work for everybody or for myself, but it'd be good to do that. Will it happen? We'll see. I am willing to push on that and just like do it myself and maybe solicit some feedback, but I don't know how much people want to care about that and then want to participate in it outside of just like nodding and saying yes, do this or don't do that.
Yeah, I think, again, it's like it was a little bit of work to do that. So, and I know that you posted a ChatGPT for Michael Levin's work that he's created. And we talked about making a custom GPT for the lab, but that's not happened yet. So we don't have that,
Well,
unfortunately.
it's well, that's since you mentioned him, well, I don't think I have the link here. Somewhere on the internet. Oh, yeah, no, it's on Twitter, and it's somewhere on my profile too. I'm just gonna call it Twitter. I don't care. Is Levin has created and released this sort of analogy bot where you type in a quote and it translates it, like you could type it like a philosophical quote and it translates from like computational biology to the context that would fit the quote, even though the quote's not about computational biology or something like that. Speaking of Levin bots,
Oh, I know. It's translating between different fields. So it's going from developmental biology to some other field, and
so I mean, we could do that. We could look into something like that for lab 2, which is, um, maybe that would be a really cool. Maybe that'd be a fun project for somebody, like an intern to do also. So, um, I'll just add that to my list of projects. Oh, also my admin hat. Like, I don't have anything great to show for this, but I am really thinking about better ways to do essentially like call for proposal, call for submission, call for interest, and like having almost like a ticketing system, both sort of internally and externally. I think internally would be more fine grained, like, hey, we need someone to do this, and externally would be a little more. We would like to have an intern that wants to, hey, does anybody want to make a Orel bot, Orel lab bot? Like, here's how Levin did it with theirs stuff, and other people, but we know that right now with the name. What would you do for us? Like that would be a mutually beneficial, fun kind of thing to do. And I think outside of that, I've been thinking quite a bit about making the systems because I am systems for ticketing basically because I think that's administratively what will help out. Yeah.
So what do you mean by ticketing system for, like, how would it?
Something like we used to have something, ideally, in the ideal Blue Skies version, something much better than what we used to do, the GitHub project boards. Something ideally, and it may not be ideal, it may just be Notion again, like a Notion project board. Something like it could be a task, but ideally it would be a system that incorporates both a public-facing advertisement for those things, like if we want an intern to come in or something, if we want a specific project, public-facing, or hold on a second. also take notes. Public facing for those for like drawing, soliciting interest, but also internally just so we organize. Okay, like I am like a little more clear way to demarcate projects and activity and like leadership in them. Like a space I would love to have. I don't know how tenable it is, but like sort of I don't know, some kind of ideally a nice graphical interface you could easily sort and say, okay, here's the different tasks by this group or type or whatever or level. Almost not quite like a jobs board, because I'm, or like Indeed or some website. Because I don't, but something a little more structured that's sort of like public facing, but also again, I think public facing might be more coarse grained and static, and just bigger, and then internally a little finer grained and detailed. Like here's like here's a opportunity that we need to do to push this project forward, something like that. I mean, it's all basic. You could say project management stuff, like make project management, put a little more time into project management and communication clarity. But then it's like, how do you do that? So
Yeah, would that require building something unique, or is there something that we can use, or how do you envision
I
it?
mean, it could totally be done in Notion. It would just take a little more effort and polishing for it. I'm hesitant to bring in, we don't want to get the farm or the zoo, the tool of zoos, the zoo of tools to be bigger and bigger and like off, go to this website to see this. Like I'm hesitant to do that, but it's there are ways to do it. Like we could just use Notion. Notion might be a great solution because you can, it's not SEO friendly or like web page friendly, but you could just make a public version of the Notion pages that have the information for public facing things. Yeah.
Okay.
I guess, I'll start from there and see there's other options.
So, yeah, that's great. So a lot of things there, a lot of kind of a administrative push and sort of a end of the year review push, and I think it's always good to stop and think about where we've been over the past year. So every year for the past couple of years, I've been doing these summary videos or summary talks, and it's always good to see how much you've done in the year, because you oftentimes forget, and there's all this stuff that just happens, and then it gets kind of pushed down, and then you end up at the end of the year like, what did we do? And you remember about half of it maybe. And so it's good to have a recap of everything, kind of enumerating everything, and then thinking about how that can push you forward for the next year. So we've had a lot of stuff this year, even though it doesn't seem like it. Not as many conferences, but then we put out some papers and we've done some things, and definitely, having the meetings themselves are part of this, lab activity. So we've done a lot of things in the meetings that are, maybe not as celebrated. So we'll have to go through that as well and see what we can find. I've been continually trying to mine the videos for the meetings and see what's there, and make sure that we have a good accounting of all that, because I think that's, if we can keep that sort of in our minds, we can kind of move forward with them. And that goes for the Cognition Futures stuff as well. We have a lot of things in the meetings that we've done that are, maybe something we can move forward with. But again, we're pushing that down, and, maybe we're forgetting about a lot of it. So I think like the Cognition Futures, we have a Cognition Futures talk on the YouTube channel, but it's not, it's kind of like a broad overview. It's not like a recap of activities. So I think it would be good to have a recap of activities. It's kind of like, this is what we've done this year. And so we've done a lot. And, even if it's not like papers or presentations. It's still like we've covered these topics, we've had these discussions, and so forth. That would be good.
Yeah, I agree. Like we've covered a lot of ground from relative Fuchs to Varroa's descendants to some other topics in like related phenomenology to now coding metaphor stuff. It's been a robust journey.
Yeah. Great. Thank you, Jesse. I did see your note about a lot of the administrative stuff you wanted to tackle, but I'm glad you were able to lay it out a little bit more. So yeah, any other updates, Amanda or Morgan?
I can go ahead.
Okay.
I guess I don't have too much to add to that. It definitely, yeah, although we don't have many, like, I don't know, deliverables from the Cognition Futures group. Every meeting has been like really, really good, really enriching, and we've covered a lot of ground. So I'm really glad that most of them are recorded. I'm excited for the cybernetics. I don't know if if it's an offshoot of Cognition Futures or what, whatever heading it falls under. I'm excited to get that going next year. Morgan and I met this past week and talked about an interesting paper. We've been talking about like nonlinear time series measures and just like EEG research in general. I'm learning a lot from my job, but it's still very like, I wouldn't say what we're doing is like state of the art anything. So it's been really enriching to look at a variety of other EEG papers. So that was good. Yeah, I think that's it. That's it for me.
All right. Thank you. Yeah, the cybernetics project is, I guess it's an offshoot of Cognition Futures, but it's also something we've been doing a long time. So it's kind of, I guess it would describe it as like an interplay between the two or something like that. It's just like, when do you put some effort into it? So it's like we did a little bit of work for a while, then it was dormant, and now we're kind of picking back up, but we're kind of bringing in some influence from the Cognition Futures stuff. So, which is good because that's probably how it's going to get moved forward. So, thank you for that update. That was good. Yeah, I saw that paper in the Slack, and I'll probably talk about it in the meeting here because it looks pretty good. Morgan, are you available to talk?
Hey there, sort of.
All right.
Excuse me. I'm still having breakfast here.
Yeah.
Yeah, we well, I really enjoyed our meetings too this week in terms of the Brett paper and the Sterling Laughlin chapter and just the discussions about those. And really enjoying meeting with Amanda and talking EEG papers. I think we still got to cover this Emery Brown, Earl Miller. Well, they've got 3 papers together. This is kind of looking at anesthesia EEG, right? But I think it'll be a good lead into the Selena Adesoy, Morton Kringleback approaches that are getting at like spatial temporal harmonics of the connectome and talking about this kind of traveling wave approaches and things like that. Especially interested in resting tissue. Resting tissue oscillations and what is physiology there, and that leads kind of back to the cerebral organoids electrophysiology the original Wojtek and Moitri 2018 paper, and how that's, I mean, I know that they're still working on things. And I've seen Allison Moitri post some things about his latest bioreactor in space. So that'll be exciting. Because just to remind people that there are interesting age effects that seem to come out from cerebral organoids in space, accelerated aging, and but that leads into the other interesting thing this week, which was meeting with a second meeting of the Biopunk Society, which is the community biolab here in San Francisco. And they have, just nice to see, a group of biotech professionals wanting to get together and make a community space like this. And Elliot Roth, who's kind of leading this, or his point person on this, has experience in a lot of other cities. And it was nice to hear that he's actually taught courses at GenSpace NYC, which is kind of the one of the most professional ones I've seen, and I've actually done some of their courses remotely. And like they previously had people from NIH and NIMH doing cRNA analysis with R or something like that, like a weekend course doing that, and so there's a lot of work that or a lot of data that's available. I've posted in the organoid channel, and it does seem like the Stanford school is no longer a winter school; it's a spring school. So, but the applications are already in or already done, but that'll be in April. So still hoping to see what they cover, but I posted a new video like just 2 weeks ago. Katerina, who we've met with, did a video with like Maxwell Biowells or something like that, and talking about their IoT cloud service with live culture.
Okay.
And what they've got there. So I think it's really interesting what we could do just remotely,
Right, yeah.
and perhaps, what I'm hoping is that we can bring an electrophysiological, understanding and analysis pipeline that they're not necessarily focused on because they're doing so many other things. And that would be one thing we could add to their kind of cloud service. So, but I also like the idea of trying to go through and summarize some of the computational psychiatry and brain organoid work for the year as a wrap up.
Yeah.
There's probably some other things if we go through I like pushing the boundaries videos. Highly recommend those to anyone that seemed really relevant to any kind of Levin discussion.
Yeah. Yeah.
Oh, sorry,
Right.
and one. I mean, there's probably a couple other things that I'll chime in with, but Amanda got in touch with John Griffiths, and he has a. So this was one of the final references in this Montreal electrophysiology and PCI of anesthesia paper, where he talks about he does this great study using computational modeling to understand what's happening with with TMS pulses and why you're getting larger or smaller responses from the pulse. And he said that he's actually got this all packaged up as a Python package, and I can't remember the exact acronym, but basically it's supposed to be pronounced Hobbit, and it's Hobbit or there and back again, which and it's because it's using recurrent, it's a recurrent network.
That's funny.
Yeah. I think he put a lot of time into that acronym. So, but I'll post a link to some of the stuff he said. And also another great meeting with Paris, and just in terms of the things that they're interested in and working on, but also some people who joined us that again are familiar with John and working on some of the Neurotech X projects, but allowing you to do experiments at home and collect data using consumer wearables.
That's really interesting.
Yeah, and so John's always led that particular Neurotech X project, which it was previously called EEG Notebooks, and now I think they're calling it EEG Experiments, which is a lot more appropriate. It's a lot clearer. But Ore, who joined us, also has like an app and web service that looks really really nicely made, and very very seamless, or very easy for people to be collecting data and tracking it from themselves. And anyway, I'll again put some more links. That was great.
Okay, yeah.
Yeah, thank you.
Amanda, did you want to make some comments on that?
Well, I shared a link to one of the papers that Morgan and I will probably discuss. Emery Brown and Earl Miller are the two authors doing this research. That's the link I shared.
Okay.
And then I guess Jesse, I don't know if you decided what you want to do with the consumer grade like EEG set and whatever the other one is, FNIR. But yeah, I'm very interested in getting my hands on something like that.
Yes, that should be a thing. I'll talk more about that later.
I can definitely, if you need just equipment, just let me know.
Okay, cool.
Yeah, that's great. Sounds like some really interesting stuff. So yeah, I can go to the Slack and look at some of the things that you posted. What channel should I be looking for?
In terms of the organoid stuff, obviously there. I see there's a couple of things that be nice to mention from events discussion. That's the link to the videos. Brain Hack Montreal is happening, and there's a couple opportunities at Genelia that people can apply for before the end of the year, or I think into January.
So let's see. Brain organoids is here. Let's start up here. Let's see. We'll start at the beginning of the month. We have the Can We Build an Electrobioreactor? That was a talk that Morgan posted.
That's the group that's making doing open source protein design, and so I thought that was interesting given the recent Tarasky talk that they were doing, for tissue engineering, they were using magnetic fields to get kind of fibrous growth. And so I was curious what this was, much more like electrocatalytic but it was still interesting. It's a great group in terms of their open source protein design.
Good. This is the Biopunk Society launch party. This is in San Francisco. So this is the opening of their facility, correct?
Yeah.
This is Aim Ahead. So this is an artificial intelligence machine learning consortium to advance health equity. So this is looks like a group that's involved in this sort of pushing this sort of stuff forward. This is an article from Boing Boing. Scientists make tiny robots out of human cells and they repair neuron cells. Well, this looks like a Michael Levin thing with some collaborators. So this is something they call anthrobots. So like every time they had Xenobots, which was Xenopus based, cellular robot. Now we have anthrobots because they're human cells. So let's see if I can pull this up. So this is the anthrobot uses human cells from the trachea that form into clumps called organoids. So they're just taking a cell that is amenable to this. They then found a way to encourage cilia, hair-like projections, to face outwards on the organoids, which were then used by the organoids to move around. So this is a very short article, but they have a video here you can check out. Yeah. And then I guess Amanda and Morgan chatted about this. Yeah. Yeah, this is, I think we talked about this before.
Yeah, whenever, I don't know, part of it just comes from me not understanding how these things work, but it definitely, some of this just elicits like I don't want to say fear, but just like, like maybe like a fear of the unknown and like using, like only having a partial knowledge of how this technology works. And then when it's like, when the discussion is about like therapeutic uses in humans, it's like, I don't know.
Oh yeah, it's very much a thing. Yeah, to like there are a lot of people who are very. They used to have problems getting stem cell research off the ground because a lot of pro-life people would say that this is an abomination, and it is pretty contentious. I mean, it still is, but there was always this sort of.
Well, yeah. It was specifically just because they were drawing from fetal tissue or from fetal blood draw, or initially.
Yeah,
right.
Yeah.
Now it's
Yeah.
It's kind of just superstitious at this point, but
yeah.
but yeah, these are I still love Levin's term, agential matter.
Right.
Yeah.
and biagential meaning like it's not us, it's doing its own thing.
Yeah.
and it can be we're showing healing, but we really don't know what the consequences of this would be when productized or potentially weaponized.
Yeah. So that leads to a lot of bioethics questions and or fear mongering. So, it's I don't want to put those in the same category, but that's sort of the landscape. And so and then this article here, which is so this is the article.
Yeah, that's just a link to the the actual.
Yeah. So this is Motile living biobot self-construct from adult human somatic progenitor seed cells. So these are progenitor cells from the trachea. They put them in this environment. They grow these little biobots. So this is these are genetically wild type cells. Anthrobots are introduced here. A spheroid-shaped multicellular biological robot with diameters ranging from 30 to 500 microns and cilia-powered locomotive abilities. They have cilia on the cells, or on the I guess they're on the organoid, or on I guess they're on the organoid. Each anthrobot begins as a single cell derived from the adult human lung and self-constructs into a multicellular motile biobot after being cultured in an extracellular matrix for 2 weeks and then transferred to a minimally viscous habitat. So these anthrobots have diverse behaviors with motility patterns ranging from tight loops to straight lines. And we talked about this with the on Wednesday with the Sterling and Laughlin article, which was the book chapter, and they talked about like single cells having these sort of cognitive like behaviors. And this is again these motility patterns. Cells often exhibit these, and the question is, how's this interpreted? Is this like a cognitive type behavior, or is this just something it's generating as a consequence of having like motor proteins and things like that? And is there sort of prediction? Is there, how is that supposed to be characterized with respect to intelligent behavior? So they kind of observe these different types of diverse behaviors. It's similar to some of the things we see not only in E. coli and in paramecium, but also in C. elegans as well. And then with speeds ranging from 5 to 50 microns per second. So the anatomical investigations reveal that this behavioral diversity is significantly correlated with their morphological diversity. Anthrobots can assume morphologies of fully polarized or wholly ciliated bodies and spherical or ellipsoid shapes, each related to a distinct movement type. So this is interesting that the shape of the morphology corresponds with the movement type. So it's like this idea of embodied movement, I guess, because it's the body shape is, and so anthrobots are found to be capable of traversing and inducing rapid repair of scratches in cultured human neural cell sheets in vitro. And then they talked about the microenvironmental cues. And so this is basically just controlling these cell populations, getting them to grow into these spheroids. Here you see this is the organoid, this is the tissue position. Here you have deep layers, shallow layers. These have some functional significance, I assume. But this is what happens, we talked about in the Sterling and Laughlin paper that you have single cells, but then you have this transition to multicellularity. And this is something that's well understood in evolutionary biology as like a transition of life. So there are a whole bunch of things that go along with multicellularity. There are also these experiments in yeast, which are eukaryotes, but they're single cell eukaryotes, but they can actually form clumps. And there have been a lot of really interesting experiments where we've had these yeasts that have kind of straddled the boundary between unicellularity and multicellularity, because a lot of their behavior exists in these clumps, and their behavior is different. And they form these different shaped clumps that people have tried to characterize. This is a similar thing, but doing this with organoids, where you have these clumps of cells that are organoids, and they have different movement behaviors. And so this is the biobot paradigm. So it's interesting. They do mention here that they transfer these biobots into a minimally viscous habitat, and there's there's an old book and some older articles. I don't know how active this line of research is, but there's this whole idea of life at low Reynolds numbers. And a Reynolds number is a parameter that characterizes the sort of the viscosity of the fluid environment. So a lot of single cells, whether they live out in like a liquid environment like a lake or an ocean, or whether they're inside the body, they exist in this flow of water or this flow of liquid. And so the idea is that you can have different regimes of turbulence in the water. It can be sort of laminar, which means there's not really much viscosity. There can be like a high turbulence or the properties which can disrupt the movement of the cells, or there can be a high level of viscosity, which means it's a very thick liquid. And so all these things really play a role in how the single cells behave, their mode of behavior, their mode of movement through that liquid or that medium. And so, this is something when you get down to the size scale, you have to think about. But it's important at larger size scales too. And, like even if you have like large fish or maybe birds in the air, they all have to go through this medium, and they have this sort of, they have to deal with the dynamics, the hydrodynamics or the aerodynamics of their environment. And so they have to, they have to adapt to that. They have to form different ways of moving through that space, through that medium. And so I find this interesting that, there's this, and by necessity, I think this focus on, oh yeah, this focus on like the environment of the organism, and they talked about that in Simon and Laughlin, or in Sterling and Laughlin, but I didn't. Yeah, I mean, I wanted to make that clear that that's like a really that the people have used that mode of investigation to sort of get some insights into very small organisms and even larger organisms. I'll have to pull up those references because they're interesting, but it's pretty obscure stuff. So, at least for this group, I mean, if you're a biophysicist, you probably know about it. But so yeah, Jesse says it would be great to try to summarize that conference somehow, but in a useful way. If people watch any of those talks, maybe we can compile our views somehow. Yeah, we again have done the shared notes documents, but what did you want to say, Jesse?
Oh yeah, just that was for the pushing the boundaries one for a while ago. But yeah.
Yeah, that would be good to kind of summarize that.
I'm reflecting in the background, like there's certain in the project management sense, there's certain like classes of activities that I want to restructure. We'll talk more about that later.
Yeah. I'm just writing something down here. Yeah, that would be good. And then Jesse says the viscosity of the medium is the question mark of the message.
Like I think there's something to that, as much as that's sort of an ironic quip. Like I don't know what it would be, but I'll have to I'll have to go to Levin's analogy bot and see what I can.
Oh yeah, that would be great. Maybe the viscosity of the medium is the friction of the message being movement.
Maybe.
Okay. Well, let's go back to the Slack. So that was good. And then we have this event here, QSB 2023. So this is, what is this, Morgan?
Sorry, let me switch back here. QSB.
We can open it up, I guess. Well, this is the winter school. So this is.
Oh, yeah. So that was the meeting on quantitative system biology.
Okay. Yeah. All right.
There's a couple meetings further down that are more organoid specific. That was very, very general, but really nice. High level overview of a bunch of kind of almost biophysics again.
Yeah.
But from a biophysics, it's systems bio. But there's a great Cold Spring Harbor meeting that's coming, and linked to the human brain organogenesis workshop. That's further down there.
All right.
Well, some good stuff in terms of
Oh, this one here.
Yeah. So that's going to be in the fall of next year, and it's got lots of great people, and then that seems like the one to go to see the latest and greatest from all the labs working on this. And then the Stanford workshop would be, to learn more about the. But it's also Sergio Pasqua organizing that Cold Spring Harbor meeting.
Yeah. I don't know. The Stanford meeting is where.
It's right underneath it. Yeah.
Oh,
sorry, it doesn't have a.
this
right
one
underneath
here,
the
yeah,
Cold
that's
Spring Harbor
Stanford,
one.
yeah.
Yeah.
Okay.
It's right there.
Brain organogenesis workshop. So this is
Yeah. Yeah, this is more of the learning, or like grad students, postdocs can apply for this, and I've wanted to know what their training material looks like, or what do they do for people. But I think if they're not going to make materials available, I think this is exactly what UC Santa Cruz is also trying to do with their live cell culture lab. So,
yeah, definitely.
Yeah.
So yeah, this is great. When we go up to this, let's see, we were at this. We had this meeting and we talked about. Right, so then this is the energetics of embryonic development. So this is this one here. Is this part of this school that we just talked about?
Yeah.
Okay.
Yeah, that was just one of the particular talks that YouTube had pointed me to, and then I was like, oh, what's this part of? And there's like 20 or 30 talks.
Yeah.
Yeah. It was just like pushing the boundaries, where it's like,
Right.
you got one recommendation, then just see like, this is 20 videos. Yeah.
This paper is interesting, or this article, I guess. Scientists stimulate mini brains to discover early developmental issues in brains of people with schizophrenia. So this is the guide to a paper, right?
Well, yeah, as well as for me also a reference to a particular group. So this is a Lieber Center at Johns Hopkins, and it's there's not a lot of groups working on this. And so when somebody's doing something, and one, it's interesting to see who's getting involved, and then two, it's interesting to see what's their take on the approach. What are they trying to push? And at this point, I think it's still a lot like Madeline Lancaster describes, where it's everybody's still kind of trying to perfect a protocol.
Right.
And, get something working. And a lot of it is still focused on trying to reduce the heterogeneity, or the the abnormalities of the artificial system. So there's stress indicators and all sorts of other abnormal cellular activity that that your protocol's trying to reduce.
Right.
But everybody's interested in this because it allows you to develop brains with particular genetic profiles, and see how those profiles differ from others, right?
Right.
So especially with something like schizophrenia, where we know it's heritable, right? But it is not Mendelian, right?
Right.
And so you've got a wide range of risk factor alleles that seem to be involved. So we really need, we need to grow brains at scale, or grow tissue at scale, so that we can look at the interactions between hundreds of risk factor genes, right?
Yeah.
And so this particular group, or Lieber Center, and Santa Barbara, I'm forgetting the last author who leads that, but the Santa Barbara Center is also a part of the Brain Engineers Santa Cruz group, and they were doing more electrophysiology. So I don't know if I'll drop a link to that particular group paper because it's actually been published.
Okay, that's good.
Yeah.
So this is unlocking the secrets of the brain's dopaminergic system. Is this a very similar type of thing, or were there
Well, this is the really exciting. So this is from Noblich, the PI of the first cerebral organoid paper.
Yeah.
So in Austria, Vienna, and what Noblich got here, which is super exciting, is like one, it's the brain's dopaminergic system. To quote, make the dopaminergic system, he's making 3 tissue types, right? So he's essentially doing 3 organoids and then linking them.
Yeah.
Right? And so again, it's like it's going to be artificial.
Yeah.
But it's really exciting in terms of actually being able to make quote full systems, with all the caveats that come with it.
Yeah.
But
I see. He's got different organoids that are projecting into one another. Is that?
Yeah.
Okay. Wow.
Yeah.
That's good.
Yeah. So and like again, not surprising from the people that got this whole thing started.
Yeah. Well, that's great.
Yeah. So it would be really worth digging into. Like again, like not all these groups share.
Yeah.
But Vienna in particular does, and and they're really keen to establish themselves as a great center for this. So it'd be really exciting to see how much they would share in terms of again, like what's needed to get the systems and the different organoids to grow together.
Okay. So this is, I guess, the Brain Engineers group work. Cowan and Kriegstein, Diversity and Evolution of Human Cortical Progenitor Cell Types. So this is talking about the diversity of cells and how to kind of incorporate that.
Yeah. I'd like that because he's actually based in San Francisco.
Okay, yeah.
But it was nice that from their website they make RNA sequence data available. They've got some data resources that they make available that would also be great kind of intro projects to start with to start looking at some of the computational biology pipelines that you would need. And here I found the COSIC lab here. Yeah, here's the. So then just this drop in one last paper there at the bottom.
Yeah.
So this is again looking like Wojtek and Mutri looking at organoids and their electrophysiology. In particular, what they've added in terms of the high density CMOS microelectrode array.
Mm-hmm.
And it's nice here because Sri Nagarajan is the electrophysiologist that I can recognize in the authors list there, and he's also up here at San Francisco.
Oh, good.
So he's the head of the biomagnetic imaging lab. So it'd be interesting to talk to him about are they continuing that work and what his thoughts are in terms of the utility of this kind of preparation and for understanding just cortical circuits or canonical cortical circuits and things like that.
Okay, yeah, so let's see. This is the method of the year here. We're going to talk about this in DivaWorm this coming Monday. This is modeling embryos. It's a paper that they had, or they had a series of papers, but this is the method of the year. So this is basically modeling development in different ways. So there have been these embryoids, or these human embryoids and these primate embryoids. I think we talked about them in this meeting a little bit a couple months ago, but they're these models of human development, like early human embryogenesis, and other types of cell models. But we also have computational models of development, and so there's a whole series of papers on this. So this is the Zernica Getz lab where they've done some of this work. So there were 2 landmark papers in 2021 that reported in vitro models of the human blastocyst, which is an early part of human development in the womb. And then there was another team that reported a human perigastrulation model, which recapitulates key events such as the formation of the amniotic and yolk sac activities and the early formation of the brain, which is early neurulation. So 3 is, let's see, Liu, Yu, and Liu. These papers here. There's also Jacob Hanna, I think, is involved in this, and Zernica Getz is also involved in this. So there are a bunch of authors that, I think that's 4 and 5. But 4 and 5 are 2 studies that were published this year reporting human stem cell derived embryo models that can recapitulate embryonic events up to day 14. This is where, you have, it isn't really much going on yet, but you start to see sort of the formation of tissues. So this is, really kind of getting at some of these issues in parallel with organoids. Now this is this one here is, I think this might be the one that I had in mind as well. The evolution of embryo models. So this is a paper that kind of goes through some of these models and talking about, there are some ethical issues that are involved with this, obviously. And this kind of goes over the different models that have been put out there so far. We've worked a lot with mouse, but also with human. So a lot of times we've used mouse as an analog of human, but it's not, really human or a primate even. So we want to use human models, but we want to do this ethically. And so this kind of goes through some of these, some of the state of the models, and then some of these associated ethical issues. So this is an interesting 3-dimensional stem cell-based embryo model guide, just showing some of these things where, they're able to generate blastoids, which is a blastula, the gastroloids, which is gastrulation, which is the stretching of the embryo, ET embryos, ETX embryos, and just where they're expressing different things. You have these mouse embryoids. You get trunk-like structures, which are sort of the beginning of the notochord and the spinal cord. Neuronal organoids, which are just kind of generic neuronal structures. PASEs, which I'm not familiar with, a post-implantation amniotic sac embryoid, and then integrated human embryo-like models. So these are the full embryo-like. And they use the term "like" because you don't want to use the actual term. So a lot of times in like stem cell biology or cellular reprogramming, they'll say something is a blank like model, like a neuron-like model. It's not a neuron. It's not a embryo. It's not. Yeah.
Yeah, but I mean, it's important to say that it's not just that's not just lawyer talk.
Right.
That's it really is speaking from this is not what we're using this term. I mean, it's again like Alison McTree saying like we need to stop calling these mini brains.
Right.
This isn't a brain. Like, and from a biological perspective, this isn't a brain.
Right.
And I think the point is that even though we're growing a neuron, it's missing all this context and environment, and we don't really know how to artificially turn on everything correctly. So you get again, you're getting all this strange molecular activity that makes it. I mean, that's why they'll talk about it being stressed or being, it's almost like it's sick.
Yeah,
Yeah.
It's more of next gen sequencing talk, because when you look at the gene expression profiles are often very different in a lot of ways.
Yeah.
But they do have
Yeah.
the morphology, which is interesting.
Right.
Yeah. So yeah, that's gonna. I'm gonna talk more about that in vivo this week. So, and then we have a number of things here: Seattle Hub for Synthetic Biology. We have Open Targets, which I found interesting. This is a public-private initiative to generate therapeutic targets based on genome scale experiments and analysis.
This looked like a really nice resource.
Yeah. And then this is the paper we just talked about, the functional neuronal circuitry and oscillatory dynamics.
Yeah. But it was nice to see that video. There is down bottom is the Katerina and her PI.
Okay.
Just talking about their work, and which is just a nice overview, and it's we should get back in touch with her. She's probably graduating soon.
Oh yeah, that'd be great.
See what she's planning to do next.
Okay. Did you want to go through the computational psychiatry channel as well, or?
I don't want to take up too much time. Let me just see if there. There was a great. There's some progress on large language models and what they're calling differential diagnosis that's interesting. There's so much to cover in large language models though. In general, this just seems like one of 40 different application areas that the big companies are following. Their teams are just so big.
Yeah.
They're going in so many different directions at the same time, which is valuable.
Yes.
So the fact that they are working on math and physics and writing novels and doing all these other things is only going to come back and help with issues that they're having in other domains like this. I pointed out hopefully we'll see the talks from the Precision Psychiatry, Personalized Neuroscience, and Future of Mental Health Treatment that Stanford did. That's Lee Thompson organizes that. I think that was it. And then just Yal Niv, I forget where I posted that, but I might have put that in Neuroscience Networks or.
Maybe.
Yal Niv has a new paper that seems really relevant to maybe Cognition Futures.
Okay.
I
But
put it in Cognition Futures actually.
Did I put it there?
Maybe. I think I know the one you're talking about.
Okay. Yeah. States as goal-directed concepts.
Yeah.
An epistemic approach to state representation learning. It's generic, but it's also her, and she's like one of the greats in computational psychiatry.
Yeah. Yeah, actually, I was thinking of covering that today. I have a bunch of things that I could cover, so I'm gonna take a look at what we have in there and see where we want to go. I think I'd like to cover a couple of the papers we talked about. But yeah, that's great. Thanks for all of that. That's good stuff. And I backed those up, so we will have those in an archive. So, but we also want to kind of again take things from the meeting and move forward with them. Not just kind of we tend to like talk about a lot of things and then they get so any comments from Amanda or Jesse on
nothing outside of I really liked in the back. I think I'm spending a lot of today's meeting in the background of like lab administrative hat or land or whatever, and thinking like, how do we there's a lot of things here that I like to tie into moving some other things forward, and I'm just thinking about that in the background. But yeah, it's quite interesting, and yeah, I'll leave it at that. If you want to cover that paper later, that'd be great. I don't have a lot of like. Oh yeah, well, I wanted to ask separately, like, what did you want to do the rest of the lab meeting today? I guess I know that papers is one of them.
Yeah, I do have some papers. I think we'll go in maybe in depth in one, and then we'll go into a topic. So I mean that unless you want to present on something or talk about something for a while, you can do that first, and then I can get into that.
Just I won't present anything, but I think it's become clear that I think there's a bit of a stepping stone project that I'm going to do and talk about that's related to everything we've said, and still in my lab administrative hat is if you have any for right now, and I will post something about this in Slack or write up something somewhere, but just general like the I'll share my screen. That might help a little bit. Let me close this.
We also did want to talk about submissions, remember? So
yeah, well, it's related.
okay,
It's sort of the other side of the same coin here. Yeah, and we can go through that. We probably should do that next. Let me move away from this noise for a second. Okay, there we go. We can go through that. I guess. Oh, I might briefly mention this too. There's a really interesting question, like identity and mathematics and history thing. I think I'll just mention that and say I want to follow up on that later. But just to put it there, let's go through this after I do this, which is basically I'm looking for citations and references that I can use to not actually put together a paper, but that I can reference at least about admin and lab management stuff. I know Levin has a paper on like he has something about managing a lab. And I want to review that again. I've been spending a lot of time this week thinking about Bradley's vision paper for the lab. Like I've been kind of meditating on that quite a bit, and just these other ways of doing stuff. Because I realized, like, I think this meeting unintentionally, I've had a little bit of like clarity, and something's clicked for me in the background about some of the work to push some of the ideas forward is at an administrative level, and it's something that's been unconsciously there for me, but I'm consciously putting it together now. So I guess I'll just like I will write up a call for it, but like if you know papers, like put them somewhere in the chat. There isn't really a great channel for it. There's actually a decent channel in the Discord, but it's kind of hard to navigate to. But you can put it anywhere. You can put it in general or Cognition Futures or other chat. I don't care. Just like tag me. If you think of something, just tag me or DM me, because I know we'll think of papers over the week. But that's something that I want to do. So I will construct this better and make it much easier to interface with later. I don't know if you have any questions or comments before we do the other things.
No, I don't have any questions.
But we can go through this then.
Yeah.
1 second. Okay. Yeah. So. I don't know if there's a few things I wanted to add to the list. I'll share my screen again in 1 second. I can maneuver. Okay. There we go. Yeah, I'll just do this. Whoops. I don't know if you want to go through any of these things or other updates. I know this date is now January 20. I think the 23rd or the 26th. I can't remember. So NIC has been extended. But yeah, anything you want to go through any of these other ones before we add something new? I'll add something new here. Let me prepare it.
Yeah. I don't really see anything that we need to update. Some of the events, I don't know if people have been following NeurIPS, but it's. I guess that's happening this week or this coming week. I'm not really sure. Kind of spread over like 2 or 3 weeks, and I don't know what the online output is. But I've been catching up on the learning on graph stuff that I got. This is on YouTube, so you can do that as well. It's just a lot of topics, that are. I'm gonna have to synthesize it a little bit more, but it's really interesting stuff. That's a fast moving field. There's a lot, of course, like we talked last week about the NeurIPS meeting. That's they've got some interesting workshops. Maybe we can try to follow up on some of that. I'm going to try to do probably not this week, but a learning on graphs recap for this year. At least my thoughts on it, because there are just so many topics that they cover, and it's just like, how do we get like the things that are important out of that? And then that's something that could be used as a guide for future work. I do actually. I'm very interested, and I think I mentioned this a couple of times. Doing something with the computational psychiatry stuff. I don't know if that's like a paper or what, but just kind of like characterizing what's in the channel or the kind of the emerging viewpoint that is kind of being represented in the channel there. So I don't know how Morgan feels about that.
I would love to do that. It's I need something to focus that.
Yeah.
And I do know some, excuse me. I do know some people that be a great reason to get in touch with them and ask them about just ask them about stuff, and where we could go with it as well.
Yeah, that would be great.
Yeah.
Do you mind if we put like just kind of a thing on the board here?
Please.
All right. Yeah. Why don't we put in why don't we talk about? Go ahead.
Well, I was just going to say, kind of related to the discussion about organoids and politicians, but I met with Jamal Bowman.
Oh, okay.
And he, his PhD work was on the educational issues that arise with adverse childhood experiences.
Yeah.
And which was also the focus of Nadine Burke Harris's the woman who became the first California Surgeon General.
Right.
Set as her 10-year goal to reduce adverse childhood experiences because these have known links to mental health and mental health issues in later life. That there's nothing that would be more to some degree more bang for the buck in terms of just reducing costs, increasing better care, better quality of life, etc. And anyway, he would love kind of like a white paper of the Child Mind Institutes, what they're doing, what that allows. He sits on the Science and Technology committee, and would love some summary of what's happening in new directions in psychiatry.
Yeah.
But with that kind of focus. And it somewhat relates to the healthcare discussion that we had previously too. Or yeah, I'd be really interested to see what Jesse has to say about that.
Yeah.
Guess I'm muted.
Yeah.
Yeah, I don't know what we call it. I just not a name kind of catchy, but I don't know what you want to call it. But yeah, like there's something here that's a bit like something just changed. A bunch of stuff just popped up from the past. I'm not sure why. But yeah, like the mental health or med and health tech thing in a direction that comes psychiatry. That's all like I don't know what to call that, but yeah, like I really want to spend time thinking at like a back end that I've been talking about the whole time, how to arrange some stuff, but then specifically like what are these like just like draw a circle around create arbitrary boundaries just to say okay in this space we want to do XYZ. So like yeah, let's like the computational psychiatry specifically. Anything else we could turn into some collection, we should do that. Anything else about that before we talk about other things?
Well, I guess, I don't know if this is a place to put any of our year-end recaps too, because it's something we'd like to do. So maybe we could do that. Yeah, year-end recaps. And then thinking of maybe at least one video, and I'm going to do this. I'm going to make a end-of-year recap for the lab. And I'm going to try to get it out by the first of the year. And, we're adding to that YouTube channel or the YouTube playlist with the open house stuff. So we have open house 2022, and that's, it's a mix of like updates from last year and like project updates. And then we're going to have another update for this year, and then maybe some more project updates or some more sort of project discussions. Again, this is stuff that, it's good to have a good recap of things just to see where they sit. And it does help you like think about the issues, I think, because when you like have to sit there and you're forced to put everything down in like a slideshow, you really get to appreciate what you've been doing. So that's good. I would say Cognition Futures, just the lab wide, and then what if people want to contribute to this, you're welcome to do so. And I'll put it in the channel in the playlist there and everything.
There's a few other things I wanted to add too. You can keep talking about that if you want. I'm just going to.
I think that's pretty much it. Again, I just think it's it keeps things tractable. Like if we just kind of think about like, especially like new directions in computational psychiatry. So I mean, it would be like an exercise in trying to distill what we had in the channel, thinking about what the issues are that are relevant to psychiatrists. Like, one way to approach that is to say, we have, in psychiatry, there's some open issues and we can apply computational methods. And these are some of the things that are going on. But then we can also talk about maybe our own sort of perspective on that, or, what we could contribute to that. I don't know the totality of the field. I'm not as immersed in this as Morgan. Yeah.
Sorry, I just wanted to say a great way to get some ideas on that I think would be really relevant for the lab is the I should have posted this, but Guillaume Dumas gets a what's it called? Cross Labs. Cross Labs AI is this. I think it's a nonprofit in Japan.
Yeah.
Yeah. Anyway, they had Guillaume talks for about an hour and a half, and just a couple days ago. And why I mention that is just that he has such a focus on social physiology, social neuroscience, and I think there might be something there just in terms of Cognition Futures or some of the VR work or etc. That you might have ideas off of. But again, it would be great to not necessarily decide what that is and talk to some people that are really working in the area, like Guillaume, like Reed Montague, Virginia Tech, and well, Yalni if she has time, and maybe is it John Murray? Anyway, some others that we could learn from. Use it as an exercise to get more, learn more about the directions people are going.
Yeah.
So from all of this, I'm seeing like I feel like what I'm leaning towards is I think I might try to create some structure and organization of some of these projects, and then basically talk to relevant people. Like for a lot of the computational stuff, I think I'm just like having a conversation with Morgan, because what you're saying is there, and like I have to in my own mind put some administrative context to it, and then come back and get what you're saying, put it into it, and make something with it. And I think that's how we're going to build it out, because I'm excited to do it, but like I was like, oh, I have to. It's like I want to cook, but my pan isn't ready yet. It's like I have to get the pan ready. So I want to come back and like I think I'll just like request specific things. Like I want to talk to Morgan. I want to talk to Amanda about it. Also Bradley, and I want to talk to myself too. But I'm excited about this because I see where to go. It's going to take some structuring. Okay, yeah, and if any, like I want to make a space so that way we can just dump, and it can be here. Like if you have ideas about this topic, I can share the link. If you want to just write whatever about it, you can do that. I'll put it in the chat if you want to do that or whatever. Okay. But like I will try to prompt, once hey, let's talk about who to take the inspiration from, where to go, and all this stuff. So yeah, I think it'll be good.
Yeah, so then I see something here, existential hope. What is that?
This is related to like some Foresight Institute stuff. I think last week Morgan mentioned that in Vision Weekend, and I didn't talk to Logan directly, but I saw some of his social media stuff, and it was really cool. Logan Dr. Collins. It's great to see him. It reminds me of like Logan did a brain emulation talk in Princeton and Vision a long time ago. So when Avery and Angela and I are all that fun time that we often reference. And I don't even know if I ever finished writing my like retrospective for that, because I keep referencing it. There's like a half-written blog post somewhere, which you can say about a lot of things. But anyway, it was great to see him. Like Logan again was talking about whole brain emulation and other stuff. But looking at that, I saw some other things on their website. I guess maybe I can shift to just to show this here. Yeah, this will work, I think. Like I was looking at Foresight Institute events in general, and I saw this X-Hope thing. I'm like, oh, what's that? And it's about very I'm sure I think about Logan as a champion of essentially existential hope. Um, and I always appreciate his um, attitude about that. And then I looked at this other stuff. There's a hackathon and a workshop and podcast, but like the hackathon looks really cool. I don't think I'll be able to go to it. That's quite in person. I don't know if it's going to be digital or not actually, but look into that. But it's kind of all AI centered, but I think it's a nice collection of people trying to do things in that space. The course looks interesting, in particular because it's sort of a co-creating and I think applications are still open. The course will start in February. You get together virtually, and like this looks interesting. Like I could see that being a fun thing to do. Still figuring it out, but I just wanted to mention it here as something that we might pursue.
Yeah.
Or try to do something in. Not saying the lab has to do it, but if anybody's interested in, we could get into the course somehow.
So what is it like? What is involved in that? Like in terms of participation?
Outside of that, sure again. Outside of this, I don't really know. I don't know if it's like going through a series of lectures or here it is. Okay. You kind of review. It looks to be 8 weeks, and you're reviewing these topics, and then you're having these discussion groups around it. I think there is sort of a vision showcase
Okay.
that you show at the end as like a deliverable, and of course you get your reward and your judges and all that stuff. But you're supposed to learn along the way strategic methods for forecasting and red teaming and all these things like that. So it feels like kind of a course / semi hackathon.
Yeah.
But I guess deliverable oriented in this way, which maybe isn't the main purview of the lab, but it kind of touches upon something. And is it probably a good crowd that I should try to participate in more? So
yeah, let's follow up on that. So that's in the list as well. Things, right?
Yeah, I'll put the link in here. The application appears to be open. I don't know when it's closing. I don't think you would date for that, but yeah,
Okay.
that's what it is.
That's great. All right, I think that's. Now I want to move to some papers. So let's see. So the first paper here is this. This is the states as goal-directed concepts. So this is something that it seems like it's probably relevant to Cognition Futures because we've been talking about states and sort of the more of the philosophy of like goal-directedness and learning and things like that. And so this might be useful here. It's also useful for thinking about learning in cognitive systems. So let's go through and see what's there. So this is Yael Niv and colleagues. This is the one that was in the Slack. Our abstract reads, our goals fundamentally shape how we experience the world. For example, when we are hungry, we tend to view objects in our environment according to whether or not they are edible or tasty. Alternatively, when we are cold, we may view the very same objects according to their ability to produce heat. Computational theories of learning in cognitive systems, such as reinforcement learning, so that's where they're putting their marker down for kind of an example of a theory of learning, use the notion of state representation to describe how agents decide which features of their environment are behaviorally relevant and which can be ignored. So this is where you have a world and you have a space that you're trying to learn, and you don't just, you learn things, but then you kind of build maybe a hierarchical model of things that are more important than others. Maybe they're affordances. Maybe they're things that are more salient. But you do build this model of, things that you pay attention to or you focus on or you tend to prefer. So this is important actually for learning how to find food or how to select things in a cluttered background or whatever. So this is good that we have this kind of, way of extracting things out of the environment. But of course, there's a process behind that. So computational theories of learning, such as reinforcement learning, use the notion of state representation to describe how agents decide which features of their environment are behaviorally relevant, which can be ignored. Again, that's if you go back to some of our open source meetings from the summer, we did some, we did some discussions on reinforcement learning and how to build a model there. So one of the things you build is you build states, and you have to represent things as states, and you have to evaluate these policies, and they reinforce a state that you're in. But state is kind of a hard thing to define because it could mean anything. It's usually some unified concept, but what does it really mean to update a state? What does it mean to form a state? And how does that get sort of added to or reduced over time? However, these approaches typically assume ground truth state representations that are known by the agent. So this is what I'm talking about with respect to ground truth representations. So if I have a representation, I kind of assume that it's obvious or that there's a ground truth to it. But really, maybe the agent is just making up states. Maybe the agent isn't really even using states in the conventional sense. Maybe they're just heuristics that are sort of cobbled together, and that's how you get these changes over time. So the idea of state is really, I think, a heuristic more for the theory sometimes than for what's actually happening in the brain or in the agent. So what's assumed is these ground truth state representations, but also reward functions that need to be learned. So, in reinforcement learning, we talk about reward functions. If you pick the right policy, you get rewarded. It furthers your learning, and that's preferred. So you pick that strategy for learning, and that contributes to the state. That contributes positively to the state. So these reward functions, and you can think about this in genetic algorithms as fitness functions. These are things that are kind of created by the programmer, or in like a biological context, maybe just kind of assumed when you're like doing the analysis. But if you're actually an agent doing this sort of learning, you don't think about a reward function or a fitness function directly. It's just kind of a thing that results from your actions. So if you pick something like an affordance or something that's salient, there's a reward or there's a fitness increase, but it's not really thought of at the time as such. It's just kind of like the obvious choice, or maybe the choice that's made. Here we suggest an alternative approach in which state representations are not assumed to be veridical or even predefined, but rather emerge from the agent's goals through interaction with its environment. So this is the idea that all of these abstract kind of reward functions and ground truth states emerge from the environment. So, the constructing a state representation, it's always a representation by the agent. They represent a state, whether it's an actual thing in the world is not actually the point. The point is that the agent sees it as a stable state in which to sort of guide their efforts of learning. And so this is where you have this, this sort of emergent state, or these emergent reward functions that kind of come about during the course of learning. We illustrate this novel perspective by inferring the goals driving rat behavior in an odor-guided choice task, and discuss its implications for developing, from first principles, an information theoretic account of goal-directed state representation, learning, and behavior. So this is basically they're arguing, this was at NeurIPS 2023, so this is actually from the Information Theoretical Principles and Cognitive Systems workshop. So we're getting some NeurIPS output in the meeting here, so good. So this looks like a pretty ambitious paper, and basically making the argument that a lot of what we're kind of, when we build a model of reinforcement learning or something like that, we're just kind of creating a model that isn't necessarily, it's kind of a summary of the biology or a summary of the behavior, but it's not like if you approach it from the standpoint of the agent, it's not really, that's not the way an agent would think about this. So they kind of start with this idea of concepts being the building blocks of mental representations. So, there's a large literature of mental representations. Concepts, of course, are sort of the building blocks of that. So you're building on concepts. You're building actually on stimuli, and then you build concepts, and then you build these representations. Oops. Okay. And so you have this, this sort of model that you're building to, but it's not something that's there a priori necessarily. How do animals, including humans, form concepts, and why do they form the particular ones that they do? And so this kind of goes through the philosophy of concepts and some of these things. To explore this hypothesis, we develop a formal framework for describing goal-dependent state representations and illustrate its application by inferring animals' goals for empirically observed behavior in a well-studied odor guide and choice task. So this is where they assume a setting for an observation action cycle, an agent receiving an observation from its environment and subsequently performing an action, then receiving a new observation, and so on. We denote by O and A the set of possible observations and actions respectively. An experience sequence, or experience for short, is a finite sequence of observation-action pairs. So this is where we have H notation equals observation and action, and observation and action, and so on. And so this is this time series. In non-deterministic settings, it'll be useful to consider distributions over experiences rather than individual experiences. So this is where you just have this, you measure this time series, you get a distribution of experiences, and then you kind of think about that distribution, and you can analyze the distribution, like the idea being that the organism has a distribution that they sort of accumulate, and they draw from that distribution. And then they, instead of having like this, focus on individual experiences being, one experience being important. That's one way to model this as a probability distribution, and it's very Bayesian in a way, because you're assuming that there's the agent is using a probability distribution. I don't know, maybe that's not the right answer, but that's the way they usually do this in modeling. Following bowling, we define a goal as a binary preference relation over experience distributions. So you define a goal as this sort of preference relation. So you're drawing from the distribution, you say, this part of the distribution, this distribution is better than this one. So that's the goal. For any pair of experience distributions, you have this notation that says that experience distribution A is weakly preferred by the agent over B. So you have this preference between different distributions, and that's where you get with respect to goal G. So that's how you get your state. It's basically a distribution of different experiences, maybe for different kinds of things, and you get this, preference in any one goal. So, any distribution is relative to a goal. If you have a different goal, a different distribution might be preferred. When both of these conditions that they define a pair hold, A and B are equally preferred with respect to the goal, and we observe that the relative, preference of a goal is an equivalence relation, so it satisfies a number of mathematical properties. So there's reflexivity, symmetry, and transitivity. So your distributions are, reflexive, symmetrical, and transitive with respect to the goal. Therefore, every goal induces a partition of this delta H into disjoint sets of equally desirable experience distributions. For goal G, we define the goal-induced state representation, S sub G, as the partition of experience distributions into equivalence classes. It induces this, which is this state representation equals the partition of disjoint sets over this G, which is the goal. And so you have this sort of relationship. So now they introduce this odor-guided choice task. So O, which is from up here, this notation O, which is the observation, is these odors, left odor, right odor, free odor. There are delays, and there's an amount. So you have spatial representation or spatial information, you have temporal information, and you have quantitative information. So you have all these sources of information available to the agent. And then you have A from up here, which is the actions. So you map O to A. And with A, you have these actions, right poke, left poke, wait for Q. We define an experience for this task as a sequence of trials. So this H, which is up here, is defined as this series of observations and actions. H here translates into LO, LP, LD, WC. So it's like you have these different elements of O and A defined by their initials. So these are just different types of observations and actions. And this is a linear representation, so there's no, we've talked about before, if you take a time series of actions, is it cumulative and do you just get a linear output? Or is it nonlinear in some way, or you get an accumulation of things and it causes things like neuronal avalanches or phase transitions or so forth? And so far, this is a linear model. But this is basically going through sort of an example from an odor task. So yeah, we have this here, and then we have, we weight them. So this is the weighted sum here. And this just weights these things by their importance. And then this is where we get this output here. We get these parameters, these beta parameters, which determine the relative weight of the corresponding difference between action or observation counts in determining the animal's goal. While our formalism is general with respect to the form of the goal, this particular goal family allows us to monitor preference for earlier rewards, larger rewards, side biases. And so this is stuff that's like, where they're preferencing the past or predicting the future, or, kind of assessing different types of magnitudes of things. So if you have a larger reward or an earlier reward, you like kind of weight your experience towards maybe later things. It's just different heuristics that the agent might use to sort of get a sense of what these are. And so then there's this goal alignment coefficient, and for a given experience, you have to measure the average increase in a goal value for each trial in H. So this is getting this sort of model of how a state is constructed. So really this is drawing from experience. It's building this state sort of over time. And you can see some of these state trajectories at the top here of figure 2, where you have simulated states and empirical states. So you can simulate this as a computational model and then observe this in mirroring model. And then this is where you have goal alignment coefficients at the bottom, which is where you optimize the weights and random weights in the model. So yeah, and then that's pretty much the paper. There's some good references in here. And so this builds on this value complexity trade-off work. There's also motivational influences of visual perception. Let's see. Neurocomputational mechanisms underlying motivated seeing, a goal-centric outlook on learning. So there's a lot of literature that's relevant to this as well.
there's a computational modeling one that she has there, 2021. Anyway, just in terms of looking for some code to play with.
Oh yeah. Which one is this? The 2020?
I think it's close. The 2021 there. Yeah.
Which one is that?
Minimal cross-trial generalization in learning and the representation of an odor-guided. Yeah, number 21.
Okay, number 21. Let's see this one here. Okay. Yeah, this is from PLOS Computational Biology. This is probably very similar to this paper. I think probably the journal version of this, but work. But yeah, this looks good. We'll have to follow up on this.
Yeah.
Any other kind of thoughts about it in terms of its usefulness or like?
Well, the one thought I had was just thinking about the discussion on Wednesday in terms of process models versus I forget what the alternative was. Substance models.
Okay.
But just the approach again being less driven by like I don't know if I'm overusing the word emergent, but like having it come out of interactions with the environment, and again seems like it's good to put this all in the context of her other work in terms of continual learning and things like that. Like it's good to not focus in a particular task and think about the organism over time.
Yeah, I think so. This continual learning aspect, where you have, your state is actually coming from these time series observations, and you're saying, what is the? Like I said, it's very Bayesian. They don't set it up as a Bayesian problem, but if you can imagine like having a prior and then using like current updates to it. That's basically what you're doing, except you're not calling it a Bayesian model. You're kind of saying, well, you're drawing from distributions of different things.
Yeah, and it somewhat relates to the new paradigms in evolutionary theory, where it's not so much survival of the fittest as the learner, the best learner,
Right.
the one it can take a change in the environment and learn from that,
Right.
and be more adaptive.
Right. And also, there's the idea of not necessarily survival of the fittest, but kind of survival of the good enough. So oftentimes we think about like evolution as being this selecting, as selecting the best, but sometimes the selection pressure isn't as strong as you would like, or sometimes there's actually neutrality in the environment. So you have this survival of like a range of phenotypes and a range of genotypes that are, pretty good, but not like the fittest. And so a lot of things are, promoted into the next generation and so forth. And so,
sure, yeah. That came out a little bit. I forget if it was in the Sterling and Laughlin, but that the motile behavior should be random. It's right. Like you don't have any sensors, so you're a harder target if you're just moving randomly.
Right.
Yeah.
Yeah, that's important. Like in bacteria, and there are different types of random strategies for movement. I know we talked about this a long time ago in the group, but there is like this Brownian motion which you can observe if you put like one bacterial cell under a microscope and you look at it. It does this Brownian motion. But in other, like in Daphnia, which is a water flea, they have different types of movement where it's like super Brownian or super stochastic, where you have like these jumps, and then you have, of course, levy flights, which are these longer sojourns that it will take around a space. So you get these, what they call super diffusive movements. So like diffusion is usually just like kind of Brownian motion, but diffusive movement is like these exponential movements where you have like 1/f type movements. So like they'll maybe move around a center and then jump to another point and move back to that same center or move to a different center or whatever. And that's I guess that would be sort of maybe non-random in a sense. I mean, it's not like just kind of exploring your space without any sort of pattern to it, but yeah, I mean, so you get stuff like that. And yeah, so it's interesting how you can model these kind of different behaviors and given different sensory inputs or environmental inputs, and how those things interact. So
Yeah, and of course, oh yeah, go ahead, Jess.
So just to bring in the lab member who's in the lab but not always here, and by that I mean Avery. Avery, we've talked about allostatic kinds in the past, and that's still a thing. But there's also a kind of Avery's looking a lot at you might just like generic stimulation and why like some of what Avery's looking at now might interestingly fit into sort of like why an agent may like in the context of levy flights, like why and how long would one want to stay in a certain area, and then where would it want to go next? And I'm just mentioning that sort of as a reference in case I come back here and look in the future, because I think Avery's doing a lot of work that kind of gets at sort of the trying to consider, or I don't know if model is really the right word here, but why in these different sort of like I was taking some notes, and I would like to leave a bit more future about this, but like the different strategies for motion, you mentioned Brownian motion, stochastic, Libby flights, all these super diffuse movements, and it's like I think some of the why and and I don't know about how, but some of the why I think ties into some of Avery's sort of deeper theory work currently, and particularly at a human level, sort of some of the drivers as to like social components for why those drivers are, or like social environmental components. Not a concrete thing to say, but more like a footnote for other things later.
yeah, okay, yeah, that's great. Morgan, did you have anything else to say before we move on? Or?
Well, just that how much of her computational psychiatry work is based on this kind of policy, reinforcement learning theory.
Yeah.
Yeah, and so this isn't just it's a wonderful use of cognitive science to inform patient not diagnosis so much as just differences. Like these are the tools that she uses to find the different strategies that individuals are using.
Yeah.
And and that's that makes up the real meat of her computational psychiatry work.
Yeah, I'd be very interested in looking more at that specifically. I don't know anything, but like I was having a conversation earlier today with a neurodivergent person about their own about like being a patient on that affected by it, and I really like to see like clinically or at this level, what people are trying to do to push that specific thing forward. Yeah.
And again, to just link it back to the work on dogs, was like, again, imagining, designing, I don't want to call them intelligence tests, but learning strategies and behavioral strategies used by all sorts of organisms, and making them broad enough so that you can get this profile, and then you can then give people feedback or tools to work on particular things that they're um, struggling with, or they find as something that they want to work on. It's just so valuable, and yeah. So that's like really, and how that, like we said before, how that feeds into obviously educational issues too.
Yeah, I think Amanda's interested in that too. And I think that's this. There's a project to happen here. I don't know what it is yet, but in that space, there's something to do.
Yeah. One more thing I want to talk about before we move on. We talk about like how do we get these distributions, or kind of have these distributions as agents, and then we have to make decisions from them. So we have to evaluate the different distributions, which I think is one is a pretty big assumption. So like it's almost as much of an assumption as that we have discrete states that we act upon. I mean, because you have to, now
Yeah.
we have models from like psychophysics on things like magnitude estimation. So in some of these experiments, you'll have people like, there's like Weber's law where you give people, you flash dots in front of people and you say, can you distinguish between two arrays of dots? Is there a difference between the two arrays of dots? And you'll, flash different arrays of dots that are very similar in terms of their count of dots. And like, you want to see how fine-grained distinctions people can make. So if you flash two arrays, one on the left-hand side that has 100 dots and one with 1,000 dots, people can make that distinction of magnitude difference. But then if you flash two arrays, one with 100 dots, one with 101 dots, people probably aren't going to be able to make that distinction. And so then the question is how what's a difference that you need to make it so that people can notice that difference in magnitude. And so that's one thing I think about when I think about distributions. If you were to train an agent on some sort of task like that and just be able to see how they can like make distinctions of things in the environment, that actually might be a way. I guess that does have like an assumption of a distribution, but it's actually just kind of an assumption of being able to detect a difference. I don't know if that's like how that would work out in a model, but that's kind of one of the things I was thinking about when I was thinking about like distributions and comparing distributions. I guess I'm not comfortable with the idea that we build these distributions and we kind of evaluate like two bell curves or something, because that seems very like this is how I've learned how to think about things in terms of operating on the past or something. This is what we do. I just don't really think that that's how we do things. But
you see, you see so many of the kind of computational psychiatry, well, not just computational, you see a lot of the cognitive scientists use these kinds of gambling tasks.
Oh yeah.
Right. But why are they using gambling tasks, right? Is because they have probability, right?
Right.
It's like they're explicitly, like it's like a variational task in the sense of variational Bayes, right? It's like we're using known distributions, like we're giving these particular choices known distributions, right? And going in. Again, I think Niv's work is great at least trying to make that more realistic, or
Yeah.
like they're dealing with it because that's the modeling tools allow them to then decompose it, right?
Right. Well, yeah.
And so it is a real problem. But you'll see in some of her computational modeling papers where they're trying to combine one or two, like two different, like you're talking about some sort of like perceptual decision making, but then like combine that with some associative learning or something like that.
Right.
Like, or as you saw in this particular thing. And certainly, as I said to her, like I just want to see more of those. I want to see more, more simulations, and make it, to make it concrete and see, how we can bring in new or more realistic, scenarios.
Yeah.
But I mean, I'm just looking at this one paper here. Humans combine value learning and hypothesis testing strategically in multidimensional probabilistic reward learning. Right?
Yeah.
But I bet a big part of it is that they've got the task set up with particular distributions.
Yeah. It's fun to think about. Like, now it looks like Amanda had to go. See you Wednesday. Thank you, Amanda. Yeah. So yeah, I'm gonna go down. I'm gonna do the other paper real quick. We have about 10 minutes left. So why don't we? This is the critical like brain dynamics. I just wanted to mention this paper and kind of go over it a little bit. I mean, we can talk about it more later offline or next week, but I just wanted to kind of give it a, kind of look through and see what's in it. So this is from the Journal of Neuroscience this year. It's actually just published. Critical like brain dynamics in a continuum from second to first order phase transition. So this talks about there are different types of phase transition. And we're talking about critical like brain dynamics. So we're talking about brain dynamics that are not linear, that have this sort of sudden transition from one state to another, or from one mode to another, or something like that. So this is something that it uses neuroimaging, MEG, and EEG. So but the model is interesting as well. So the abstract reads: the classic brain criticality hypothesis postulates that the brain benefits from operating near a continuous second-order phase transition. Slow feedback regulation of neural activity could, however, lead to a discontinuous first-order transition and thereby bistable activity. So this is where you have these different types of phase transition that the brain operates toward near this continuous second-order phase transition, but you could end up with a discontinuous first-order transition that moves things into another state, or have this, where you can have bistable activity. Observations of bistability in awake brain activity have nonetheless remained scarce, and its functional significance unclear. Moreover, there is no empirical evidence to support the hypothesis that the human brain could operate flexibly near either a first or second order phase transition, despite such a continuum being common in models. So we have these models of like change in terms of brain states, but it's really hard to find support for these hypotheses, to find, exactly how these things. So it's like, using a model that is a good metaphor, but it's not necessarily, you can't really demonstrate it conclusively with the data. So that's been the problem with a lot of these models. So they use computational modeling, and they find bistable synchronization dynamics that emerge through elevated positive feedback. So this is where you get increased positive feedback, and occur exclusively in a regimen of critical-like dynamics. We then assessed bistability in vivo with resting state MEG in healthy adults. So they built this computational model. They were able to sort of replicate this kind of behavior. Then they looked at bistability in resting MEG in healthy adults and in EEG in epilepsy patients. This analysis revealed that a large fraction of the neocortices, which are the neocortex regions exhibited varying degrees of bistability in neuronal oscillations from 3 to 200 hertz. So this corresponds with their modeling results. The neuronal bistability position are positively correlated with classic assessment of brain criticality across narrowband frequencies. So the brain criticality hypothesis is that you have brain activity and you have this accumulation of like noise and things like that, and then at some critical point there's a transition into a different state, and the idea is that the fluctuations, in oscillations, different forms of noise can tip the brain into this new state. We talked about the sand pile model as being sort of an analogy for that, where you have this time series, you build this pile of sand, you keep dropping grains of sand on top of a pile of sand, and eventually you get to a point where that pile has some sort of displacement, because when you keep adding grains of sand, there's some point at which there's a failure mode, and that part of that mound of sand fails, and there's a landslide. And so in neuronal avalanches, you typically get these, accumulations of noise or other kinds of oscillatory dynamics that shift it to a new state, kind of, stochastically. So it's hard to predict, but you can observe it, and that's the idea there. Excessive bistability was predictive of epileptic pathophysiology in the patients, whereas moderate bistability is positively correlated with task performance in the healthy subjects. These empirical findings thus reveal the human brain is a one-of-a-kind complex system that exhibits critical dynamics in a continuum between continuous and discontinuous phase transitions. So this kind of talks about, this brings this idea of different types of phase transition, and let's go into like kind of maybe talking about some of these phase transitions and what they kind of used for bistability here. So there's been this brain criticality hypothesis that's been around for a while. If you go to Beggs 2008 or Beggs and Plenz or Dante Chialvo's work, they talk about this quite a bit. They basically posit that brains, like numerous other complex systems, operate near a critical point of continuous second-order transition between asynchronous and fully synchronous phases. And so this is where they kind of lay out this idea of the neural avalanche. This would lead to moderate mean synchronization and emergent power law spatiotemporal correlations, and yield many functional benefits, such as maximal dynamic range. So you can actually measure this in the power law spectrum. If you extract power law spectra from these time series, you can see that there are, some, very few epochs of very intense activity, and then a lot of epochs of lower activity, and you can, plot that out and get a power law exponent, and, it can tell you things about like the dynamics of that time series. So there's a whole sort of methodology around this. Conversely, operation far from the phase transition would be associated with inadequate or excessive synchronization of neuronal activity, both of which are incompatible with healthy brain functions. So there's this idea of synchronization where, you don't have enough synchronization in terms of like, incoherent brain activity, or excessive synchronization in the case of epilepsy, where you have these, regions of disorder, basically. And this is a way to characterize these types of regimes. So, there's this balance of excitation and inhibition, which we need to characterize, and they kind of do this. So their canonical model for bistability is this Hopf bifurcation. And so this is where they kind of use this model for representing bistability. You can use this, and it comes from physics, so you can use this as a model. In this reference, Fryer, they use this as a kinetic model of gene regulation, and suggested the universality of the canonical model for bistability across a broad range of biological systems. So this is the Hopf bifurcation the equation for it. So, they looked at this in gene regulation, but they were saying that you can apply this to brain activity or any other set of a wide range of biological processes. So they talk about, different types of noise. So you have, high state dependent multiplicative noise that can result in erratic jumps between a low and a high amplitude attractor, which is driven by Brownian noise. So there are different ways you can model this. Assumptions of noise in neural signals. You have a lot of different sources of noise. Some of it's external, some of it's internal. There's, noise is not always a bad thing. It can be a good thing. It can, be part of the dynamics. So, but you have to characterize it properly to get a good result. We hypothesized that different combinations of two parameters resulted in either supercritical or subcritical bifurcations, and they talk about the different types of Hopf bifurcations in the Fryer paper, but these are associated with underlying continuous or discontinuous. So a continuous phase transition is a second order, and a discontinuous phase transition is a first order. So a first order phase transition is usually associated with some sort of like discontinuous jump, whereas a second order phase transition is more of a continuous phenomenon. So this is something that I'm not prepared to explain in detail right now, but these references, Kim, DeSanto, and Kochi et al., they describe this in more detail, especially with respect to the brain. So they kind of go through, they look at bistability in a Kuromoto model. The Kuromoto model has a number of oscillators that are modeled as this array of oscillators that are connected, and they can model like different neurons behaving collectively. So you can actually get these kind of complex. You can basically model the dynamical output of the brain and some of these other higher order organizational principles. So the Kuromoto model is a generative model that can be used for studying the collective behaviors of a number of interconnected phase oscillators. And so we can look at this as a collection of phase oscillators. And so then we can move on to the data collection and then the results, which now these are the maps that they usually use for this. Actually, they show the time series here where you have supercritical, critical, and subcritical behavior, and then this bistable behavior. So you can see that there's this critical behavior, something that's subcritical and supercritical, and then that translates into like the shift between one state and another state, and then back to the original state. So you can see that you have this critical behavior that kind of corresponds with some of these shifts. So this is kind of, but this alone wouldn't be enough to sort of say definitively that there's this effect. You have these, other types of maps that show these regimes. So for different parameter values, you have kind of describes where you should expect to see these regimes. So you have this bistable zone between the critical and supercritical, and then the subcritical is back here. So basically, if something is near, sort of heading towards supercritical, that's when you start to see these signatures of bistability or a shift in the different states. And so a lot of data, and I think we're out of time pretty much, but this kind of this really gets into their analysis of brain activity and getting into some of these measures a little bit more and how they work out. So this is like extracting things from the neuroimaging data and kind of replicating this model in data. So, yeah. Now, I know Morgan maybe want to say a few things about it.
Well, just this was as Amanda said in Slack, this came out of the paper that we read this week. We covered this week on criticality measures and resting EEG predicting perturbative complexity index, or this TMS derived measure, where you are hidden people not as a cognitive task, but as purely to probe the dynamics of the system. Right. So, and as well as like a lot of the people who work on criticality are interested in um, loss of consciousness, anesthesia, sleep, seizures. When the brain goes into one of these new states, right? Or what being, and that can be subcritical, supercritical, and these other dynamical regimes, right? So the paper we covered was like propofol and xenon would take you sub or supercritical, and a ketamine infusion at a particular dosage, you would be unresponsive, but you'd have critical like behavior or critical like activity, and that they would also report dreams and that was still you might say there was still consciousness, even though they were unresponsive. But it's a really interesting area, and and I like where this paper is going in terms of everybody's trying to simulate EEG electrophysiological recordings with as much capturing as much of the complexity as possible, right?
Yeah.
And this is really important. The generative important. Sorry, go ahead.
Oh yeah, I wanted to point to this this figure we were talking about with where they had the supercritical, the subcritical, and the critical.
Yeah.
So the subcritical is basically like a lot of inhibition, or the supercritical might be a lot of excitation. So like supercritical might be like a seizure. Am I understanding that correctly? And then subcritical would be something where there's a lot of disorder.
Yeah, I mean, I would, or like, I would want to start with things like sleep stages.
Okay. Yeah.
And as we talked about, and Amanda and I talked about, and somewhat with people in Paris and Brussels, these computational models that we have for simulating EEG.
Yeah.
It's we're always trying to do resting, or we're trying to do wake EEG.
Okay. Yeah.
And all these models really, like, they should be able to do sleep stages and resting wake.
Right.
And task. But that these same, the EEG is interesting because EEG is not sensitive to the whole brain, right?
Right.
So we're sensitive to this particular cell population, and those cell populations are only directly tied to a couple others.
Right. Yeah.
So we have this kind of canonical circuit in terms of what we're sensitive to. And when we're asleep, we still, polysomnography is still the gold standard in sleep medicine. So, it's EEG, right?
Yeah.
And so this is the same area of cortex is displaying this very different behavior. And our models should be able to, our computational models should be able to switch between these. And in a explanatory or an interpretable mechanistic fashion.
Yeah.
Right.
Yeah.
And you see that. So it'd be great to follow up on that paper. Like I said, I'm following up on with John Griffiths, who did some of the best computational model that I've seen, and he's using that same Janssen RIT model that's used in SPM's dynamic causal modeling.
Right.
But is predicting the TMS response, and again, this is not just a theoretical interest, but you can use this for people who are unresponsive, if you do TMS and you're recording some electrical activity, you can get a much better estimate of what's going on internally for them. Are they conscious
Yeah.
or not? And a lot of the references that you'll see in this bioarchive paper are really about dealing with patients in the ICU, dealing with patients in the emergency room, dealing with patients in comas, and getting at even you can have spatial heterogeneity. So this particular region is not so responsive to TMS, but this region is, and that can be really informative for patient care. But the physics is definitely getting quite demanding.
Yeah. It's
but Michael Breakspear, who's one of the co-authors there, has done some nice review articles in terms of the physics underpinnings of these that I would recommend.
okay. Yeah. All right. Well, I think that's it for today. Another good meeting as usual, and let's keep this up in the Slack. Or actually, I've sort of replicated some of the computational psychiatry stuff in the Discord, in the Big Ideas Discord. So we can also discuss things there.
Yeah, I think mentioning the Discord just before we go, I think some of the project work is going to be in those Discord threads that we made for the project. What is it here? It's the project dev management, and I think stay tuned in that space, and we're probably going to tag people for discussions there.
Right.
But yeah, this is great, and a lot of fodder to think about.
Yeah.
So thank you.
All right. See you.